Hello everyone, welcome back. This is a supplemental lecture that goes along with our lecture series on photosynthesis. So this still belongs to our Theme 2 Topic 4, and today's focus is going to be looking at plants that use slightly different pathways for photosynthesis. So we're going to be looking at the C4 and Cam plants. And this we're going to see is in response to living in really hot, dry conditions. So just to go over a little bit about what's happening with the plants and some of their anatomy here is recall that in our last lecture we talked about the enzyme rubisco, and rubisco was able to either bind with carbon dioxide or oxygen in its active site. So when levels of oxygen increase inside of the plants or inside of these leaves, it's going to tend to favor what we described as photorespiration, which decreases the amount of sugar that can be produced and also consumes more energy. One of the reasons that this can happen is when plants are in these hot, dry climates. So before we look at that, let's just kind of review some really basic anatomy here of some of our leaf structure. All right, so if you look at the leaves, if you go outside today before all the leaves fall off the trees, right, you'll notice that leaves are covered with a waxy coating. The purpose of this coating is to prevent water from evaporating from this very flat surface, right? It's also there to prevent transport of CO2 and O2. So the leaves are trying to regulate to some regard the amount of CO2 that is coming into the leaf itself and the amount of O2 that's leaving. So this also, the wax in essence, prevents a barrier for them to just freely transport across that surface. So in order for water and these two gases, in order to leave and enter the leaves themselves, there are special cells that surround pores. And together, this is known as a stoma or stomata for plural. So we have these little pores, as you can see here in this picture, and it is surrounded by these guard cells that are kind of like muscle cells. If we were to kind of find a similar structure in an animal cell, they're not called muscle cells, but they are going to allow for the opening and closing of this pore. So they kind of function like a muscle would. All right, and so these guard cells are allowing are going to open and close this pore based on the environment that that leaf is in. If we look at this bottom picture, we see that in general, carbon dioxide is going to flow into the leaf where water and oxygen tend to flow out. All right, and this is because the concentration of carbon dioxide inside the leaf is going to be significantly less compared to the amount of carbon dioxide located outside of the leaf. And so by simple diffusion, carbon dioxide is going to want to enter. All right, so let's just review that real quick. I know for some people it helps to have this in a written outline form. So again, open stomata allow for carbon dioxide to diffuse into the leaf and for oxygen to diffuse out. There is a very strong or high concentration gradient that favors CO2 entering into the leaf of the plant, and this is maintained by the Kelvin cycle. So if you remember our lecture from the other day, the Kelvin cycle is where we use carbon dioxide and fix it to our UBP in the first step of making our G3P precursor carbohydrate, right? And so because the Kelvin cycle is using up CO2 as the carbon dioxide comes into the leaf and into those cells, it's being used up. So it's helping us to maintain this concentration gradient. In most plants, and a typical plant that lives in an environment where there is plenty of moisture and moderate temperatures, similar to what we might see here in Boone, you would find that the stomata are typically going to be open during the day and closed at night. All right, the purpose for this is that while the sun is out and the light dependent reactions are taking place, right, so are the light independent, right? So the stomata are open, it's bringing in that carbon dioxide, the Calvin cycle is functioning. Right, so most plants, you know, or those that live in climates similar to the ones we live in here in Boone, are going to follow this process, right? So having the stomata open during the day and closed at night. However, when conditions are really hot and dry, stomata are going to close. They have to prevent evaporation of water. And so when they close, what we see is that that also prevents oxygen, right, from leaving the leaf because of that waxy coating. And so what happens is we start to get this buildup of oxygen inside. And as we just mentioned, rubisco also will bind oxygen, not just carbon dioxide. And so photorespiration is actually going to start to take place. So this would not be a great scenario for a plant to be in long term. So if I'm a plant that lives out in the Sahara Desert or in Arizona, someplace that's pretty hot and dry, even, you know, out in the Midwest, in corn country there, these conditions are not great. And it would basically cause the plant to eventually die. So over time, plants have adapted that live in these hot, dry climates. And that's what we're going to take a look at now, is what these adaptations are. So the first adaptation that we're going to look at is known as the C4 pathway. All right, and just to clarify this random name that just showed up, the pathway that we've been talking about all along is technically known as the C3 pathway. So where the stomata are open during the day and closed at night, and you have the light dependent reactions happening at the same time as the light independent reactions. They're kind of working, you know, hand in hand then. Those are the C3 plants, or the C3 pathway. So in the C4 pathway, this is one that we're going to see in our sugarcane plants and things like corn or maize. So plants, again, that are trying to adapt to a hot, dry climate. What we're going to see is that they're going to fix the carbon dioxide to a different 3-carbon compound with an enzyme known as PEP carboxylase. All right, and so as soon as that carbon enters into the cell, it's going to be kind of captured and basically turned into this 4-carbon compound. All right, so this is different to what happens in our C3 pathway, where we are going to attach the carbon dioxide to RUBP first. Okay, so this is an additional step that's taking place. So let's see how this helps the plants kind of avoid photorespiration. All right, so again, just kind of comparing a C4 plant to a C3 plant. When carbon dioxide enters these plants, in C4 plants, it binds to a 3-carbon sugar, where in a C3 plant, the carbon dioxide binds to a 5-carbon sugar known as RUBP. So there's one difference there. The enzymes used are different, right? So for a C4 plant, they have this enzyme known as PEP carboxylase, where our C3 plants used rubisco. And we basically generate a 4-carbon organic acid through this reaction for the C4 plants. And in the C3 plants, this is where we get our 3-PGA, so that 3-phosphoglycerate molecule, right? Which is also an organic acid. All right, so how is this helping our C4 plants? So what we're going to see is that in these plants, instead of carbon fixation and the Calvin cycle both occurring in the same cell, this is going to take place in actually 2 different cell types within the plant. So the first carbon fixation is, again, to that 3-carbon molecule using PEP carboxylase. And this is taking place in a cell type known as the mesophyll cells. And these cells are located near the surface of the leaf. What happens then is that this 4-carbon molecule then is able to travel to these cells known as the bundle sheath cells, which are more towards the interior of the leaf and are closer to the veins of the leaf so that it can be transported. The 4-carbon molecules then are broken down in these bundle sheath cells to release the carbon dioxide. Now this carbon dioxide is able to enter the Calvin cycle, all right, and basically go through the same process that we saw with the C3 cells. So the normal thing that we saw with the fixation, the reduction, and then the regeneration. So that will all take place now, but now it's taking place in these bundle sheath cells. The 3-carbon molecule that the carbon dioxide had bound to upon entering our leaf, it had been released, and we're going to recycle it. So this 3-carbon molecule now returns to the mesophyll cells, where it's now able to fix another incoming carbon dioxide molecule. So we've basically masked the carbon dioxide and kind of shuttled it in to the Calvin cycle so that hopefully we're able to prevent that photorespiration process from occurring. All right, so here's what it kind of looks like in a cartoon drawing. So here is the leaf surface. And you can see here these kind of like lighter green or olive green colored cells. These are known as the mesophyll cells. And in these C4 plants, these contain the enzyme PEP carboxylase, right? So they're going to fix our carbon dioxide as soon as it enters in, and then basically shuttle it here to the bundle sheath cells where the enzyme rubisco is located. And then it's near this vascular tissue. So it's able to make the sugar and then get it into the vascular tissue of the plant so it can be transported. All right, so if we look here as a close-up image, you see the carbon dioxide coming in. We're able to have that reaction with PEP carboxylase, making our 4-carbon compound that can then enter here into our bundle sheath cell. During this process, another enzyme is used to basically separate the carbon dioxide from the carbon molecule that it had bound to. So now we have carbon dioxide directly right here by rubisco. So rubisco can pick it up and basically perform the Calvin cycle and make the sugar. So rather than having like all this oxygen that's out here, the oxygen doesn't have as great of a chance, right, of getting into this inner region here to be able to react with rubisco. So our cells here are able to prevent photorespiration by using this process, and therefore helping these plants to survive in these hot climates. The second adaptation that some other plants that are in much drier climates, such as cacti, have adapted is known as the CAM pathway. This pathway, similar to the C4 pathway, is also going to fix carbon dioxide to a 3-carbon compound to form that 4-carbon organic compound. So it does this very similar to the C4 pathway, and we'll see that it also is then going to release the carbon dioxide so that it can interact with RUBP similar to the C3 pathway. All right, so the CAM, again, is using very similar things that we've talked about with both C3 and C4. What's interesting, though, about the CAM plants is that they actually open their stomata at night and fix carbon dioxide then. So in the desert, right, it can be really hot and dry during the day, but at night the temperatures are a lot cooler, which is going to prevent a lot of evaporation from these plants. So if they open their stomata at night, it's safer for them to be able to let the carbon dioxide in, the oxygen out, and also not lose a lot of water. So the carbon dioxide then is able to basically be shuttled to the Calvin cycle during the day then when the stomata are closed and perform the light independent reactions. So I think this is one of the really, really clever adaptation that these plants have made, right? So at night, these plants, and again, I believe these are the mesophyll cells, they are collecting all of the carbon dioxide that they can, and they're also getting rid of all of the oxygen, you know, that they've built up. And so when they do this, they can store that carbon dioxide as this 4-carbon compound. And as you remember, if the light reactions aren't going on, I don't have a supply of energy. I don't have any ATP or NADPH to run the Kelvin cycle. So it has to wait until the daytime when the sun is back out. Now I've got a huge store of carbon dioxide. I've closed my stomata so that you know, I'm not losing water, and I'm able to run the Kelvin cycle and generate my G3P molecule. So pretty clever. This is keeping these cacti and other desert plants or your succulent plants alive during these really hot, dry times of the day. So both C4 and CAM pathways function to concentrate carbon dioxide, again, to increase its concentration so that it stays at a level elevated to that of the oxygen. So as long as we can keep carbon dioxide at this elevated level, it will have a greater chance of interacting, right, with the rubisco enzyme rather than the oxygen. So this is really good for the Kelvin cycle, will help us move forward with photosynthesis and making G3P. So just as a one-line statement to kind of summarize each of these pathways, the C4 plants are going to spatially separate the carbon fixation and the Kelvin cycle. So remember, we used mesophyll cells and the bundle sheath cells to separate the two processes. The CAM plants temporarily separate carbon fixation and the Kelvin cycle. This is more a spatial or a time separation, right? So temporally, sorry, I read that as temporally, right? So they are, one is, you know, it's collecting the carbon dioxide at night, and then during the day, it's going to run the Kelvin cycle. So yes, they're temporally separated. All right, and so that's just a couple of the adaptations that plants have made so that they can be more efficient at photosynthesis, depending on where they live. So what happens to the sugar that is produced by photosynthesis? So we mentioned that the actual product we're making with photosynthesis is this G3P molecule. This molecule is often used then to make glucose in fructose. And the name of that process is known as gluconeogenesis. So genesis means formation, gluco means sugar, and neo means new. So this is the new formation of sugar. And so this process will take place outside of the chloroplast there. And basically the glucose and the fructose sugar can then later be combined to form sucrose, or if there's too much sucrose, the glucose monomers will be polymerized to form starch. All right, and so starch is, again, the storage molecule for our plants. And then here's a little question for you to think about. Starch is broken down into glucose at night. Why do you think it does that? So I'm going to leave you with that question. You're welcome to post the response to this in our Ask It basket. And this concludes actually all of our lectures on photosynthesis. So if you have any questions about this process, please feel free to contact me or post a question in the Ask It basket, and we'll try to get that addressed for you. So have a great day, and I'll see you shortly.